Hello, and a warm welcome to everybody for joining us at today's webinar. We're looking forward to an exciting presentation where we will review the discovery of potent pan-KRAS inhibitors for gastrointestinal and pancreatic cancers. Speaking today, we have Dr. Gary A. Piazza, co-founder and chief scientist of ADT Pharmaceuticals and professor at Harrison College of Pharmacy at Auburn University. He is the department head of drug discovery and development, and he is also the director at the Cancer Research Center. Professor Piazza will lead us through the layers of target biology, mechanism of action of the novel KRAS inhibitors, and the preclinical data. Next, we will hear from Dr. Ivan Babik, a drug discovery scientist, inventor, entrepreneur, and co-founder of CellarisBio. Dr. Babik will provide a review of how CellarisBio's cell target engagement technology contributed to discovery and optimization of novel pan-KRAS inhibitors. After today's presentations, we'll have some time for a question and answer session. So please use the Q&A function in Zoom to submit your questions that you have throughout the webinar, and we'll answer them at the end. Now it's my privilege to turn things over to Professor Piazza. Professor Piazza, we are very excited to hear what you have to tell us.
Okay, well, thank you, Emily, and thank you for the kind invitation to give this webinar. So I'm going to be talking about a discovery of a novel class of RAS inhibitors. I'm a professor at Auburn University, and for sake of disclosure, I'm a co-founder and chief scientist at ADT Pharmaceuticals. Just some background here. RAS, it's most frequently mutated oncogene in all of human cancers. It's key driver of tumor cell proliferation, metastases, a number of other processes essential for cancer tumorigenesis. It's mutated in over 30% of human cancers, particularly pancreatic, colorectal, and lung cancer. It's activated by mutations or amplification upstream pathway components, various receptor tyrosine kinases, for example. And it's also activated as tumors become resistant to chemotherapy or radiation. It's long been considered undruggable, but recently immune-specific G12C KRAS inhibitors have been approved. So that suggests that RAS is indeed druggable. And there's a tremendous effort in this field of trying to identify, develop small molecule inhibitors of RAS at various vulnerabilities. The data we have right now suggests that the efficacy of immune-specific KRAS inhibitors is limited through a variety of resistance mechanisms, most notably the emergence of new mutations in KRAS or other RAS isoforms, or unchecked activity of wild-type RAS isoforms. So I'll be discussing this in my presentation. Okay, I'm going to be focusing mostly on pancreatic cancer. It's mutated in over 95% of patients with pancreatic cancers. The G12C inhibitors, while they're efficacious for non-small cell lung cancer, they have really limited utility for pancreatic cancer. Less than 3% of patients have the G12C mutations. As you can see from the diagram on the right there, that pancreatic cancer is it's a complex mutational landscape of a variety of different KRAS mutations. But I'm going to be talking about a highly potent pan-RAS inhibitor, which we believe would be ideal for pancreatic cancer and other RAS-driven cancers. It has very potent and selective activity, robust in vivo activity. And we see that pancreatic cancer cells are very sensitive and are not susceptible to mechanisms of resistance that would limit the efficacy or the use of immune-specific KRAS inhibitors. Just background, RAS signaling is critical for tumorigenesis. The PI3 kinase and the MAP kinase pathway are two such downstream pathways resulting from activated RAS as a result of either mutations or from activation or amplification in receptor tyrosine kinases. RAS, it's a signaling molecule. It's a GTPase. It's considered a switch on the off position. It's bound to GDP. In the on position, it's bound to GTP. There's specialized proteins for removing high affinity nucleotides, GEFs to remove GDP, or GTP activating proteins to remove GTP, to basically regulate the cycling, the on-off of RAS controlling proliferation of cancer cells. So we had discovered our lead compound ADT-007 by screening an indene library. So indenes are small molecules. The rationale for the screen was literature going back probably over 20 years now, 25 years, that a non-steroidal anti-inflammatory drug, Celandec, showed some modest RAS inhibitory activity. So through a medicinal chemistry effort, we've synthesized a large focus library of over 2,000 indines, and we screened them for RAS inhibitory activity in RAS wild-type HT29 cells, or RAS mutant HCT116, both colorectal tumor cell lines. And our lead compound ADT-007 showed a very potent and selective inhibition of RAS mutant cells, over 100-fold difference in selectivity for the RAS wild type HT29 cells. Differences in selectivity for inducing apoptosis, apoptosis in the RAS mutant cells, but not the wild type cells, and longer duration of treatment with colony formation assays. Again, we see this very potent sub-nanomolar concentrations are able to inhibit colony formation of RAS mutant cells, but not the RAS wild type cells. Focusing on pancreatic cancer cells, these are 4 pancreatic lines with different mutations in KRAS, single-digit IC50 values, low nanomolar IC50 values, and compare that to BXPC3. It's similar to HT29 in that the downstream mutation is in RAF. And they are essentially insensitive to growth inhibition by ADT-007. In this case, we're seeing approximately a 1,000-fold difference in the IC50 value to inhibit growth of the RAS mutant cells versus wild type cells. And corresponding with the mutational status, we see high levels of activated RAS. This is measured by a RAS pull-down assay, high levels of activated RAS in the RAS mutant cells, but low levels of activated RAS in the RAS wild-type cells. The compound is active in monolayer cultures as well as in spheroid cultures. IC50 values are comparable regardless if their cells are grown as a spheroid or monolayer culture. Again, longer duration of treatment, you get very pronounced complete inhibition of colony formation in the RAS mutant pancreatic cells, but not in the RAS wild type pancreatic cells. We've looked at a large number of cell lines over the years, regardless of the histological origin of the cancer cell line, or the KRAS mutation, or mutations in HRAS or NRAS. We get a very low nanomolar IC50 values. And you compare that to tumor cells with wild-type RAS, and in this case, they have the B downstream RAF mutation. They're very insensitive to treatment with ADT-007. These are done in, this is a 3 or 4 day growth cell viability assay using cell titer glow, measuring cell viability. Normal cells, for example, normal melanocytes or normal human airway epithelial cells are essentially insensitive to treatment. As you would expect from a pan-RAS inhibitor, cells with wild-type RAS, but upstream mutations, and for example, receptor tyrosine kinases or GTPase proteins, these cell lines are exquisitely sensitive to ADT-007. So the question is RAS the target? We've addressed this through our collaboration with Cellaris, but also this is another key experiment where we took RAS wild-type HT29 cells. We transfected them with G12C immune HRAS. You can see the parental cell lines. The IC50 values are 500 to 700 nanomolar. And by transfecting in immune RAS, as confirmed by high levels of activated RAS by RAS pull-down assays, the dose response curve is shifted to the left, generating sensitivity similar to cells that have endogenous immune RAS. A key feature of these compounds is their ability to inhibit activated RAS, and we're showing this under three different conditions: under basal conditions, just normal cell culture conditions, or if we serum starve the cells, or if we serum starve the cells and then we stimulate with EGF for 10 minutes. In all cases, the cells were incubated overnight with ADT-007, and you can see that the concentration to reduce activated RAS as measured by RAS pull-down assays was pretty comparable to growth IC50 values. Here we're making use of pan-RAS antibodies. So antibody, a pan-RAS antibody against all, whether KRAS, NRAS, or HRAS, or isoform-specific antibodies, whether specific for KRAS, NRAS, or HRAS. And then we're these Miapaka cells with the G12C mutation. We're exposing them to overnight incubation with ADT-007, treatment with EGF. With pan-RAS antibody, as I've shown you before, we get the reduction of all RAS isoforms, but specifically with these pan-RAS antibodies, you can see that, first of all, as you would expect, EGF has really no effect on KRAS because the KRAS is already mutated, but EGF is clearly stimulating wild-type NRAS and HRAS. And in all cases, ADT-007 is decreasing levels of activated RAS, of the endogenous mutant RAS, as well as the activated NRAS and HRAS. This shows really the exquisite chemical and biological selectivity of ADT-007. I'm just showing you a part of the moiety of the molecule. ADT-007 has a critical phenolic OH moiety. This moiety is essential for both the potency as well as the selectivity of the 007 compound in growth assays. So again, I'm comparing Miapaka cells to BXPC3 cells, 1,000-fold difference in selectivity. But if you change only this hydroxyl group and you put a methoxy group on, we lose potency and we also lose selectivity in the growth assays. And corresponding with the decrease in potency and the decrease in selectivity, and the RAS are with the phenolic OH, we see the effect on reducing activated RAS, but with the methoxy group substitution, we lose the effect on activated RAS. So a real nice correlation between effects in growth assays and effects in RAS pull-down assays, and by making just one change to the chemical structure of the ADT-007 with this ADT-106 analog. As you would expect, ADT-007 reduces MAP kinase signaling as well as PI3 kinase signaling. On the left here is KRAS mutant HCT116 cells grown under basal conditions. We see a drop in activated RAS, as I've shown you previously, but reduction in MAP kinase signaling, as well as PI3 kinase AKT signaling. On the right here is Miapaka cells, where we've stimulated them with EGF. And again, we see the reduction of MAP kinase signaling and reduction of AKT signaling. We've made use of resistant cell lines. These are Miapaka cell lines that were established to be resistant to either Amgen's sotorasib AMG 510 or Mirati's 1257 compound. These cell lines were indeed resistant to treatment with both the Amgen's G12C inhibitor and Mirati's, but maintained sensitivity to our ADT-007 compound, pan-RAS inhibitor compound. This is an important example. This is a situation or scenario where the tumor cells, this Lewis lung carcinoma cells, where they acquired two mutations, a G12C mutation in KRAS, but a secondary mutation in NRAS, the Q61A mutation. As you would expect, AMG, because of that second mutation, does not inhibit the growth of Lewis lung carcinoma cells, but ADT-007 does maintain potency in these cells with the dual mutations in KRAS and NRAS. We've done molecular modeling, trying to better understand how ADT-007 is binding RAS. So using this as a crystal structure of wild type KRAS using induced fit molecular modeling. When we do this, we get a very tight binding of ADT-007. In the absence of magnesium, and where we're removing the nucleotide, and under this conformation, the binding affinity for GDP drops from minus 15 down all the way down to -8. So suggesting a mechanism whereby ADT is binding RAS when it's in this nucleotide-free conformation, and within the catalytic domain to disrupt GTP or a nucleotide binding to RAS. So in support of that, we've collaboration with an NMR spectroscopist with expertise in that area, with recombinant KRAS. ADT-007 affects NMR spectrum of a number of residues, but in particular, these two residues are within the GTP binding domain of RAS, where we're seeing clear signal attenuation by treatment with ADT-007. I should point out that this was done under nucleotide or under magnesium-free, and where we're generating nucleotide-free RAS, or in the open conformation. We did not see any evidence of binding when RAS was bound to nucleotide. To support these observations, we've done our pull-down assays with the RAS pull-down assay when under conditions of nucleotide-free RAS. ADT-007 disrupts binding of RAS to RAF, but no such disruption when RAS is bound to GTP. Using a fluorescent derivative of GTP, MAT-GTP, we've done kinetic studies using recombinant RAS. This the blue is the binding of GTP to RAS under nucleotide-free conditions. The green is inhibition with ADT-007, and the black is where the low basal levels of GTP binding, MAT-GTP binding, if RAS is bound to GDP. So this is our model where we have evidence that ADT-007 binds RAS when it's in this open nucleotide-free state. This has been the presence of RAS in this conformation has been proven by Dr. O'Brien at University of South Carolina. He's done a lot of work with monobodies quantifying nucleotide-free RAS. But we believe that it provides a unique opportunity for ADT-007 to bind RAS and inhibit GTP binding. And it's known in the literature that nucleotide-free RAS is a non-stable, so causing an irreversible degradation of KRAS to inhibit its activation. Now, you might be asking a very important question: what's the basis for selectivity? How can one might predict that a pan-RAS inhibitor would be overtly toxic? How are we getting this selectivity? And we believe the answer lies in the chemical selectivity of ADT-007, and in particular this phenolic OH moiety. And so we did an experiment where we did an overnight treatment with ADT-007, 100 nanomolar, 24-hour treatment with ADT-007. We quantified ADT007 levels in either RAS wild type or RAS mutant cells by LC mass spec, and we see this very large difference in the intracellular level of ADT007 between RAS wild type and RAS mutant cells. You can see there's in fact, a concentration or an enrichment of the compound in the RAS mutant cells, but very low levels in the RAS wild type cells. And corresponding, we see high levels of the glucuronidated form of ADT007 in the wild type cells, but undetectable levels of the glucuronide in the RAS mutant cells. So that suggests a mechanism that could contribute to the selectivity. And there's probably a mechanism where the cells are RAS addicted, contributing to their sensitivity, but this unique mechanism of glucuronidation to render the cells, RAS wild type cells, resistant. And in support of this idea, we've quantified UGT expression in RAS wild type cells, HT29 or BXPC3, both show appreciably higher levels of UGT, the enzyme responsible for glucuronidation of phenolic compounds such as ADT007, much higher expression in the RAS wild type relative to RAS mutant cell lines. Further evidence, we made use of an inhibitor of UGT propofol, pre-treatment with or simultaneous treatment of RAS mutant or RAS wild type cells with ADT007 and the UGT inhibitor. In the inhibitor significantly shifts the dose response curve to the left in the RAS wild type cells, but no significant effect in the RAS mutant cells. So again, the UGT inhibitor is not affecting RAS mutant cells, but having a significant impact on inducing sensitivity of the RAS wild type cells to the ADT007. I've shown you this table earlier, but this is an additional lane that I've added looking at UGT expression in the RAS mutant cell lines. And these cell lines have uniformly low levels of UGT relative to lines that have high levels of UGT, and these are lines that are the most resistant. So we have 4 examples of lines that are resistant to ADT and very high IC50 values and appreciably higher UGT levels relative to RAS mutant or ADT007 sensitive lines. So this is our model where what we believe contributes to the whereby normal cells would be refractory to pan-RAS inhibitors. So basically, RAS wild type cells or normal cells would have high UGT levels. It's well known to be expressed in the colon, kidney, liver, for example, but also a variety of other normal tissues. So high levels of UGT will glucuronidate the compound. That makes it more water soluble and a substrate for MRP or efflux proteins. So I'm going to shift gears here, do some discuss in vivo studies with ADT007. First of all, I want to point out it's the phenolic OH moiety was given us grief with it's rapidly metabolized by liver glucuronidation. So to do proof of principle, we directly injected the compound into the tumor. We see strong inhibition of tumor growth. Corresponding with that, we see inhibition of MAP kinase signaling, as well as on PD-L1 levels. So this is a similar experiment where we're directly injecting the compound, but this is a patient-derived xenograft model. Again, where we do peri-tumor injection, we see strong tumor regression with ADT007. So another experiment done by our collaborators in Norway. This is a peritoneal model involving PDX cells. The tumor cells were growing in the intraperitoneal cavity. ADT007 was injected intraperitoneally. We see a marked reduction of mucin volume and a very significant difference in survival between untreated mice and mice treated with ADT007. So this obviously presents the need for pharmaceutical optimization. So we made a prodrug of ADT-007. It's water soluble, metabolically more stable. Compound we refer to as ADT-1007. This is the pharmacokinetics of measuring ADT-007 following a single dose of ADT-1004, the prodrug. Prodrug is very well tolerated. We're not seeing any adverse effects on any histopathology on a variety of normal tissues at doses way up to 175 milligrams per kilogram given twice weekly. So the compound's well tolerated, and the question is, what are we getting efficacy with 1004? And the answer is yes, we're getting very strong efficacy at dosages appreciably lower than the maximum tolerated dose. So for these experiments, rather than testing at the maximum tolerated dose, we wanted to first find the minimally efficacious dose, and it seems to be around 10, 20 milligrams. We're still, this is a pancreatic, a mouse tumor line with a G12D KRAS mutation. We see strong inhibition of tumor growth. And I should point out, we're still optimizing the dose. We're trying to figure out what's the optimal dose and schedule of administration. This is another example of experiment in a PDX model. Again, inhibition of tumor growth. Compound's very well tolerated. And see strong inhibition of tumor growth in a standard subcutaneous model with a PDX tumor. So I'm going to summarize here. ADT-007, potent and selective killing of cancer cells with mutant or activated RAS. And this is why we describe it as a pan-RAS inhibitor. Binds RAS to block GTP activation effector interactions and downstream signaling. Importantly, of MAP kinase as well as AKT signaling. And this was what you would expect from a direct-acting RAS inhibitor. We've done other experiments, which I haven't discussed, but there's a low potential for off-target effects. And I think that's evidenced by the selectivity that we're seeing in our cancer cell lines. But we've done other profiling experiments that convince us that the probability for off-target effects is very low. And certainly that's evident in our in vivo studies where we're getting a wide therapeutic window between efficacy and any effects on weight gain. I've shown you evidence that low potential for resistance. There's a variety of mechanisms that limit the efficacy of the mutant KRAS specific inhibitors, and we believe that by inhibiting both mutant RAS as well as co-expressed RAS wild type isozymes that can compensate for a specific KRAS inhibitor, we believe a pan-RAS inhibitor would have efficacy advantages as well as, of course, broader utility. Data I haven't shown you, but we've collaboration with an immunologist at UAB generated evidence that the compound activates innate and adaptive immunity, and certainly has the potential for combinations or to either improve efficacy or broaden the efficacy of checkpoint immunotherapy. We've identified an orally bioavailable prodrug. We believe this is a strong development candidate. It has robust anti-tumor activity in mouse models of pancreatic cancer without a discernible toxicity. So it's a development candidate ready for IND-enabling studies. So I'm going to finish up here. I want to acknowledge my collaborators, Dr. Chen, a medicinal chemist. Dr. Keaton has been studying the mechanism of action. Dr. Barry, mouse tumor models. Maxo Tanko, strong background in pharmacology and toxicology. I've been supported by NIH for way back since 2004. National Cancer Institute has, we've established a very nice relationship with the Stepping Stones program, and where there's some very active collaboration we have ongoing with doing essential development work for IND-enabling studies. And want to shout out to my colleagues at UAB and ADT Pharmaceutics. So I'm going to stop there and hand it over to Dr. Babak.
Thank you so much, Dr. Piazza. That was very informative and very interesting. We appreciate that. Okay, yes, now we are going to hear from Dr. Babak regarding CellarisBio's involvement in this exciting new technology. So please take it away, Dr. Babak.
Thank you, Emily. Thank you, Gary. Very interesting talk and very comprehensive, and happy that CellarisBio could be a part of it. So early on in their study, they wanted to define whether they want to determine whether the ADT-007 molecule was binding to KRAS, so direct engagement of this ligand to its potential target, because they had functional readout and they wanted to demonstrate that the phenotype that they were observing in the functional readout was through a direct engagement of ADT-007 with KRAS. And so this is where we came in and helped their study by generating a KRAS G12C mutant that was tagged with the microtag. So this is a 15 amino acid tag. On panel to the left is just an immunoblot with an anti-microtag antibody demonstrating expression of this KRAS G12C tagged version, showing that the construct expressed at correct molecular weight, and there are no degradation fragments. Then testing this in our assay complementation system. This is where we add reagents to that would bind to the 15 amino acid tag and create a fluorescent signal. So this is just to quantify the amount of protein that is there. So panel B just demonstrates that that particular tag is amenable for enzyme complementation. There's significant fluorescence detected compared to the untransfected control. So in these type of assays where we're examining target engagement, it's commonplace to identify a temperature of aggregation or a temperature of melting at which that protein is going to denature 50%, and that would be the temperature at which to interrogate ligand binding. So in this case here, applying a thermal gradient to KRAS G12C. These are lysates, so these are non-denaturing lysates prepared from cells expressing the construct. And with increasing temperature, there's a temperature of aggregation that can be identified at about 52 degrees Celsius for this particular construct, this protein. Now, testing binding at that particular temperature, so incubating the lysates with two potential compounds that they identified, ADT-030 and ADT-007, shows some increase in fluorescent signal as you increasing the dose of both of these inhibitors. Significant increase in signal suggests that there is a stabilization of that target in the heat challenge. And ADT-007 demonstrated an EC50 of target engagement of about 8 nanomolar compared to the 030, which had about 85 nanomolar. So we have been continually improving our target engagement system to try to provide more valuable data, to try to streamline the process, reduce cost, and improve sensitivity. And here's where we've started to introduce the use of real-time systems rather than using plate readers, which are static readouts of interaction. In this particular case, what we've started to do is introduce the lysates expressing the construct with the with inhibitors and immediately do real-time target engagement detection. So here is what it would look like, or what it does look like when we do a thermal profile. So this is increasing temperatures for this particular KRAS mutant is a G12C mutant. And as you can see, as you increase the temperature, you start to see a decrease in the amount of fluorescent signal over time. So this derivative of these curves can identify a temperature of aggregation of about 44 degrees for this particular mutant construct. So these are lysates expressing the KRAS G12C mutant. When we look to assess target engagement. So here we're looking at a Mirati compound 849, MRTX 849, binding to KRAS G12C. As we increase the dose of this particular ligand, what we're seeing is a separation of these curves. So we start to see an increase in fluorescent signal over time with increasing dose. Comparing that with the wild type KRAS version, there's just absolutely no binding showing selectivity of this particular inhibitor for KRAS G12C. And this Mirati compound 849 is a covalent, irreversible covalent inhibitor of the G12C mutant. So that was just one example of some of the constructs that CellarisBio works with. KRAS is a well-behaving target, works very well in these type of target engagement assays. But we've expanded our platform to look at more challenging type of targets. For example, transcription factors, membrane proteins, and other challenging targets such as UBE2N. So this is some of the data that we will be presenting at upcoming conferences, for example, AACR coming up in April. And we've improved our platform, increased sensitivity, decreased the cost, and are continuing striving to improve these target engagement strategies to assist clients. And I'll hand it over to Emily now.
Thank you so much, Dr. Babic. That was very interesting. Okay, thank you everyone for joining us today. We'll now have an opportunity to answer some of the questions that came in during today's presentation. If you have any questions that weren't answered in today's presentation, please feel free to put them in the Q&A section of Zoom, and we'll get to as many as we can. But before we get there, we'd love to ask you a question. On your screen will appear a poll question. Are you interested in testing CellarisBio's cell target engagement technology? If those of you in attendance wouldn't mind answering that question for us, we would be very grateful for your interaction. Okay, so we will now turn to our questions that came in. The first one that came in quite early in the session, and I believe Dr. Piazza addressed it later on in his presentation, but they were wondering about the efficacy in vivo. I wondered if you had something else to add to that, Professor Piazza, or do you think that was covered in the presentation?
Yeah, certainly I've presented, and I have my colleague here, Dr. Keaton, to help me field questions. So certainly feel free to ask as many questions as you like. But yeah, the in vivo data is very convincing, but we are continuing to expand. We are interested in comparing our pan-RAS inhibitor with the pan-KRAS inhibitors. The G12, the immune-specific inhibitors are in development, so those studies are underway. We're also making use of the resistance model that we've established, the Miapaka resistant line. So those experiments are undergone. And we also want to do in vivo comparisons between RAS wild type lines, for example, BXPC3 versus RAS mutant line, Miapaka. So those experiments are underway. We'll be presenting some of this data at AACR as well as hopefully ASCO as well.
Fantastic. We'll look out for that. Thank you, Dr. Piazza. Perfect. Okay, our next question. Your structure suggests the possibility of quinone methide formation and tracks with the OH-OMe SAR shift. Have you explored this possibility in your MOA studies?
Okay, I'm going to ask my colleague, Dr. Keaton, to address that question.
Hi, thanks, Dr. Piazza. We did explore that, and I should preface by saying I'm not a medicinal chemist, but our colleague, Dr. Chen, did prepare the oxidized form to address this issue, and we found that compound was actually very unstable and not durable at all. We did do our best to study that in vitro and found that those compounds were inactive, presumably because of their instability.
Thank you so much, and thank you for joining us. We're happy to have you here. Okay, perfect. Our next question is, what mice were used in the mouse models? SCID?
Yeah, we've done a variety of different mouse tumor models, immune competent models with the mirroring cancer cell lines. So those were non-immune competent. I don't believe we've used skids yet. Although Dr. Maxotenko, collaborators at UAB would probably be able to address it, but I think we've been primarily using athymic on nude mice.
Perfect. Thank you so much. Okay, and our final question as of yet, and so if you have another one, please get it in the chat now. Otherwise, this will be our final question. Except as the downstream signal of and activated by EGF, are there any ROS ligands that can directly activate ROS?
Yeah, I'm not, maybe if you could clarify the question. Yeah, we're RAS ligands. So this would be GTP. Are there any other ligands that would activate RAS other than GTP? I guess that's, if I'm understanding the question. I'm not sure I understand the question.
No problem. If the person who asked that question wouldn't mind putting a clarifying question, or you can also send an email to CellarisBio, and we can also answer it later. Oh, yes, he says that's the
Okay.
question, as you understood it, Dr. Piazza.
Very good.
So would you like to answer that question as you understood it, or?
Oh, okay, yeah, I assume the question is, are
Yes.
there nucleotides other than GTP that activate RAS? And gosh, I'm sure this has been studied in the literature. I'm not aware of any, though. I mean, certainly there's probably some promiscuity among with the substrates. The MANT GTP, for example, will bind at RAS and presumably activate RAS. Whether or not other nucleotides will activate RAS, I'm just not sure. But it's a good question. Encourage you to do some Google searching.
And it's a good answer. If we don't know, we don't know. Perfect. Excellent. We have one more question that came in for Dr. Babic now. Does microtag system measure any functional effect of the inhibitor?
No, it's only direct target engagement. So the functional readouts are typically complementing assays.
Okay, perfect. Thank you so much, Dr. Babbage. Okay, that concludes today's webinar. Thank you so much, everyone, for attending. If any more questions come up, we'd love for you to email us. We are hi@celarisbio.com. Please follow us on LinkedIn and also our website, celarisbio.com. We'd like to thank all of you who participated, our panelists and our visiting doctor. Thank you so much for answering some questions for us. And we invite you all to stay tuned for future webinars from CellarisBio. Thank you all and have a wonderful day.
Thank you.